I just want to preface this talk with this slide that shows that this webinar is actually the third of a series of 3 that we've put on this October. First webinar was basically a tour de force introduction to microRNA function and analysis by John Schaefer, who's actually on this call. The second was concentrating primarily on microRNA quantification and normalization in PCR analysis by Subbu Uramelli, who also is part of our group. And this last one by me is concentrating more on profiling microRNA expression in cells, FFPE, and serum as sort of on the road to biomarker development. So I have a few introductory slides to bring everyone up to the same point before we start talking about technical details. Many of you probably know the stories of how RNAi was discovered. In fact, there were 2 divergent phenomenon that ultimately converged about 5 years ago. Victor Ambrose and colleagues had found one small functional RNA, which was just a bit of phenomenology, which, as it turns out, was the first microRNA to be discovered. But really, the field of RNAi exploded through work of Andy Fire and Craig Mello, who won the Nobel Prize for their work. And it was almost by accident, in that people were using long antisense RNA and C. elegans as functional tools to knock out gene expression. And it worked sometimes, and it worked sometimes better than other times. And what Fire and Mello found was that in their preps of antisense RNA, when they, for example, made a new prep, one prep may have worked better than another. And the thought was that if they could purify their long antisense RNAs better, that they would make a more functional molecule. And what they found was that when they highly purified their long single-strand antisense RNAs, in fact, the potency of the prep went down. And so what they discovered, which a lot of us who did in vitro transcription in the old days would discover, is that when you linearize a plasmid and you make an antisense RNA, that there's a very small amount of plasmid that's not cut. And because of that, you make a little bit of the wrong strand of your insert of your plasmid, and this little bit of their wrong strand would find its complement in the vast majority of the antisense and make a double-strand RNA. So this minor component, this little bit of double-strand RNA, in fact, was the potent silencing molecule in their preps, and if they purified that or deliberately made double-strand RNA, it was thousands of times more potent than the antisense RNA was. And this discovery led to essentially the discovery of RNA interference, that double-strand RNA direct the specific silencing of a gene. And the mechanism for this gene-specific silencing that they were using as a functional tool for functional genomics, as it turns out, is very conserved from plants to humans. And this is where the second part of the story comes in, and Victor Ambrose's small RNA, small functional RNA, in that, as it turns out, most organisms use this RNAi pathway as a natural form of gene regulation. That short double-strand RNAs, microRNAs, are used for fine regulation of gene control. So when you look at this slide that's in front of you now, what you see is that Science magazine called RNAi the breakthrough of the year in 2002. Fortune recognized a billion-dollar breakthrough for RNAi in 2003, and you ask yourself if Fire and Mello discovered RNAi years before this, and you look at this graph and you see that there were virtually no microRNAs discovered and virtually no papers published in 2001 or 2002. Why was there all this excitement? And the reason was that Tom Tuchel and colleagues around 2001 discovered that as opposed to C. elegans, if you tried to put long double-strand RNA into a mammalian cell, mammalian cells would have a potent interferon response, and they would undergo apoptosis and die. So this was a tool, a functional genomics tool, that was restricted to lower organisms until Tom Tuchel discovered that if you took short double-strand RNA of the kind that Victor Ambrose described years ago, a short double-strand RNA, put it in a mammalian cell, you could target a specific gene. So the field of the microRNA regulation of gene expression and the field of using small double-strand RNAs as molecular tools for functional genomics merged right around 2002, and we discovered that this was all one big phenomenon. And that's what you can see once people realized that microRNAs existed in other organisms and that you could use them as microRNAs or siRNAs as functional tools, the field just exploded. So we went from the miR-based database of a handful of microRNAs in 2001 and 2002 to, for example, over 16,000 today. And in humans alone, there have been described more than 1,100 microRNAs. And if you follow that line, the number of publications track almost identically with the number of microRNAs that have been discovered, and that in 2010 there's more than 10,000 papers of people either looking at the phenomenology of microRNA regulation of gene expression or using them as tools. So in fact, this is a breakthrough of probably more than a decade, and it is certainly a billion-dollar breakthrough in that pharma and biotech companies recognize the potential of small molecules that can be used to target individual genes or target individual pathways. So this is a rough diagram of the canonical pathway of microRNA biogenesis. MicroRNAs are unique in that the genetic locus that encodes it can either be a protein encoding gene, a Pol2 gene, and the microRNA sequence is intronic, exonic, or in the 3' UTR of a protein coding gene. It can be its own transcription unit with its own Pol2 promoter, or there can be multiple microRNAs embedded within one primary transcript, all under the control of one Pol2 promoter. So the interesting thing here is the genomic organization is complex. The other interesting thing about this is it means that these microRNAs are under the control of the same cis and trans-acting factors that control the expression of mRNAs. And in fact, many of them are coordinately transcribed with an mRNA gene because they're embedded in that primary transcript. So once a microRNA primary transcript is made, the characteristic secondary structure of it is recognized by the Drosha degenerate complex. And if you think about it, this happens to a microRNA in an mRNA. So at the same time that the cell is processing the primary mRNA transcript and splicing out introns, the Drosha degenerate machinery has to recognize this structure and process out the pre-microRNA, and there's evidence that these two features are in fact linked together. This pre-microRNA is exported out of the nucleus specifically by exportin 5. It's not just by diffusion. And once in the cytoplasm, this short hairpin RNA is recognized by another complex that contains dicer and TRBP, which leaves the hairpin on one end, trims the other end, unwinds the two strands of this microRNA, and physically hands one strand or the other off to a multi-protein complex that assembles around this strand of RNA called a guide strand. It recruits an RNA endonuclease called AGO. There are several different forms of AGO, some of which are actually active endonucleases and some which are not. This assembled RISC complex then uses that 21-base guide strand of microRNA to recognize a region of homology in an mRNA. It was originally thought that region was exclusively going to be in the 3' UTR of an mRNA, but there have been examples where that region is in the coding region. And it was originally thought that a microRNA would bind to the 3' UTR and cause translational repression. But what we also know is that if that sequence is perfect homology to the mRNA, it can cause site-specific cleavage. So initially, the hallmark of a microRNA interaction was repression of translation, but multiple papers have shown that in addition to repression of translation, the RISC complex binding to mRNAs does cause quite widespread mRNA degradation and the loss of mRNA as well. The net result is that when a microRNA binds to a target, we get less protein made from that mRNA. So that's a lot of steps in this biogenesis, and when you think about microRNAs and being dysregulated in disease, there are a number of spots along this pathway that can be disturbed that will ultimately affect that interaction of the risk complex with its target transcript. And what I show on this slide, for every feature that I mention here, in fact, there are papers that describe these particular disturbances. So if we look on the left side here, we can see that you obviously can change the interaction of a microRNA with its target by changing the stoichiometry, and we can do that by altering transcription of the microRNA, the primary microRNA transcript. We can do that by changing the activity of its promoter, by differential methylation. We can change it by histone modifications. We can change it by modulation of transacting factors that interact with the promoter, like transcription factors. All of these have been described as methods by which microRNAs and their targets have had their normal interaction disrupted. On the right side, we know that in cancer, there's frequently genomic instability. There are amplifications and deletions of loci, translocations, and other events like insertional mutagenesis. And in fact, there are several very, very well-studied instances where the loss of a long non-coding RNA or the loss of an mRNA is causative in cancer, and as hard as people would try to figure out what that mRNA or what that long non-coding RNA was doing to cause, where its loss would cause oncogenesis, it finally would turn out that in fact the long RNA or the mRNA was not the causative agent, but the loss of a microRNA that was associated with that locus would cause oncogenesis. You could have disruptions in normal DROSHA processing, either by disturbance to DROSHA itself, modulating DROSHA behavior, or point mutations or other types of mutations within the microRNA transcript that changes its secondary structure. You can disturb dicer processing by a number of ways, all of which can affect the final mature level of that microRNA that would be available to interact with the target transcript. And finally, you can disrupt that interaction by disturbances to the target transcript itself. So you can lose the microRNA binding site in the target. We know that in oncogenesis, there's a progressive loss of 3' UTRs in many transcripts, which would probably mean that you're losing the ability to regulate those transcripts by the interaction of a risk complex with the 3' UTR. There are examples of alternative splicing where you lose microRNA binding sites, and of course, you can have SNPs or other mutations that destroy binding sites. All of these different methods for disturbing that complex on the bottom have been described in publication, and all of which can result in disease or disturbance in normal development. So over the last 5 or 6 years now, it seemed like a natural thing to do once we realized that there was more than one microRNA in the world, was to look at how these microRNAs were regulated. And it was discovered that their regulation was, their temporal and spatial distribution was as complex as any mRNA, and that's not surprising because they're encoded by Pol II promoters. For years, there were heated discussions at scientific meetings of whether there were less than 100 microRNAs, between 100 and 200 microRNAs, and as I mentioned, now we know that there's more than 1,000 that are known in humans, and there's probably more that we haven't discovered yet. It was natural that we look at both regions of genomic instability in cancer and microRNA expression patterns in cancer and other diseases to see whether we can find relationships between the disease and disturbances in either the genomic loci or expression patterns. And what was discovered early on was that many microRNAs were located in genomic regions that were unstable in cancer. There's a cluster called miR1292, which is frequently amplified in cancer, and the miR15A, miR16 cluster is frequently deleted in cancer. And we know that these microRNAs can act as tumor suppressors. I mean, since then we've found a number of microRNAs that are abnormally expressed in both solid tumors and in blood cancers. And there's been a lot of work that's shown that microRNA expression patterns can correlate with both the clinical biological characteristics of tumors. So you cannot just diagnose a tumor, but you can predict its level of differentiation, aggression, potential for metastases, and response to different therapies. So when all of this is said and done, clearly both academic researchers, researchers in biotech and researchers in pharma are intensely interested in looking at microRNA levels as both predictors, prognostic for disease, and also as potential biomarkers, and also as potential targets of drugs or drugs themselves. So this slide now is probably a year old. It's probably bigger at this point if I went back and did this analysis. But this is just a small list of companies that were either using microRNAs as drugs, so for example tumor suppressors, or as the target of a drug, a microRNA inhibitor, in preclinical trials. And so there are probably now 24 different targets/diseases where the manipulation of a microRNA, either to stimulate it or to inhibit it, is thought to be a potential therapeutic. And most of these are cancers, but some of them are, for example, hepatitis or HIV or high cholesterol and things of that nature. So with that, I'd like to turn and talk a little bit about microRNA isolation technologies. QIAGEN has been the standard bearer for nucleic acid sample prep for 25 years, and for microRNA, that's no exception. The basic technology that we use is the basic QIAGEN miRNeasy technology that's been optimized for small RNA recovery. And if we look at the left side of this panel, what you see is that we're doing a homogenization, a disruption of our cells and tissues in an organic solvent with chaetrope. So the product is called Chiazol, and it's not the standard that we use for miRNeasy, but it does increase the yield and the quality of small RNAs in samples, so it's the standard for microRNeasy. The aqueous phase of this organic homogenate is then bound to a column, washed, so impurities are washed away, and we elute a total RNA that's got a quantitative and qualitative recovery of microRNA. And this is done in either a spin column format for small, low throughput, or it can be done in 96-well filter plates for higher throughput. The spin columns can be automated on the QIAcube for walkaway automation of low throughput, and the filter plates can be automated on our larger liquid handling robots. There's a separate protocol for purification and enrichment of just the small RNA fraction. This is something that we do not need to do for qRT-PCR profiling of microRNAs, but it has its uses for cloning and sequencing, for enriching small RNAs, for labeling to use on microarray experiments and such. And that protocol is also included in the miRNeasy manual. But again, it's not something we're going to be talking about throughout this webinar. We actually now have a second version of our miRNeasy FFPE Kit available. There are estimated to be hundreds of millions of FFPE samples that are archived in tissue banks, pathology labs, and biomedical research labs. The hallmark of FFPE samples for nucleic acid is that the nucleic acid is typically fragmented. It's cross-linked to proteins by the formalin, and that cross-linking typically interferes with reverse transcription, and it's hard to get the RNA out of these samples. We're lucky with microRNAs because they're so small. If these samples are in generally good shape, we can get microRNAs out that we can do profiling on. We do a deparaffinization. We digest the protein with proteinase K, and that gets rid of most of the cross-linking to nucleic acids, and then we can do a DNase treatment and then do a bind, wash, and elute and get enough material for miRNome profiling from FFPE samples. The two things that will generally spell success or failure for FFPE are the age of the blocks. Obviously, if they're really old, things tend to go bad if they're 25 years old. Whether they've been sealed and any moisture has gotten into them, and also how long they were sitting in formalin before they were dehydrated and embedded. So these are These are things that affect any molecular biology experiment that's done on an FFPE block. One of the biggest things to hit microRNA research in the last few years is profiling microRNAs in blood. And if you think about blood, we know that there's multiple cellular components like red blood cells, white blood cells, possibly circulating tumor cells, platelets, other cells shed from linings of blood vessels or from tissues that the blood passes through. But really the most interesting thing is the discovery of extracellular stable microRNA in circulation, so in plasma and serum. And I'm an RNAologist from 25 years ago, and I know that there are incredibly high levels of nucleases present in blood, which makes sense, actually. I mean, you would think you wouldn't want, naked RNA and DNA in circulation. These are things you'd probably not want to have move around the body. However, stable microRNA and a stable profile can be detected in serum and plasma. And we'll talk more about the prep on the upcoming slides, and I'll show you some data a little bit further in the talk. So why is there stable microRNA in circulation? Well, this is an evolving field. There are a number of papers that have published conflicting data on this, and what's probably true is that all of these things are true. So first of all, in healthy people, there are hundreds of microRNAs that are detectable in serum or plasma that are in a nuclease-resistant format. And these have been shown to be present in exosomes and microvesicles, so you can perform an ultracentrifugation and pellet these and purify them. They've been shown to be not in exosomes, but be complexed with AGO2, and these can be brought down by immunopurification. They've been shown to be complexed with HDL, and in fact, these microRNAs can be taken up by cells at distant sites that are exposed to these complexes in blood. And then there's other. And other is some other protective protein that's not AGO2 that microRNAs in serum and plasma in circulation have been shown to be complexed with. So probably all of this is true. Why are these microRNAs there is a very good question. Some of them may be part of some signaling system in the body that we don't understand yet. Some of them are shed from cells that die. Some of them are maybe actively shed. There's published evidence that, for example, microRNAs are selectively excreted in exosomes from cancer cells to deliberately lower the level of that microRNA in the cell. And that, to me, is an unbelievable thing. And this is all very recent data, and it's all evolving, and the story will only get more interesting. We purify microRNA from serum or plasma by using the standard miRNeasy mini kit. There's a protocol available online, which we've optimized, which you can download from Qiagen.com. It's a very minor change to the miRNeasy mini kit. We also recommend a spike in control. So when we add our chiazole and produce a homogenate of the organic solvent and our serum or plasma, we add a synthetic C. elegans miR39 transcript, which is available also. And what this lets us do is monitor recovery of nucleic acid from samples so that if you screw it up later on and your experiment fails, you'll know that you lost your RNA, and it also allows you to do some normalization between samples in terms of % recovery. Circulating microRNAs are not only in plasma. So here is a table from a paper by Weber et al. From Clinical Chemistry in 2010, where they looked at the miRNome in many different biological fluids from humans. So plasma, as we spoke about, tears, urine, and we are ourselves doing work in urine, breast milk, seminal fluid, saliva, amniotic fluid, bronchial lavage, CSF, pleural fluid, fluid from the peritoneum, and colostrum. So it's not a unique phenomenon to serum. MicroRNAs appear to be in a protected format in most body fluids, and again, why they're there is probably incredibly interesting. So how do we look at these microRNAs now that we've isolated them? So first I want to briefly describe the system that we're using, which is the miScript II system. Historically, QIAGEN released miScript version 1 a few years ago. QIAGEN also is now partnered with SA Biosciences, which you'll see some of their products in upcoming slides. And we've taken essentially the best of both systems, and I think that we've advanced this to a system that's significantly better than either system was to begin with. And I'll describe that to you on these upcoming slides. So miScript II is actually two different technologies in the same kit. The hallmark of miScript version 1 was that we were able to use a single RT reaction and detect all RNA molecules, which means mature microRNAs, precursor microRNAs, and I'll explain why that's important, mRNAs, and any other non-coding RNAs in the sample. And you would do this when you were interested in looking at the dynamics of microRNA processing, when you were wanting to look in the same sample at the relationship between a microRNA and its target mRNA. It's a nice experimental tool to look at microRNA and its regulation of gene expression in cells. But more people today are interested in doing profiling of microRNAs. How do related panels of microRNAs change in different disease states, under different treatments, and even to look at the whole miRNome between samples to do biomarker development. And for that, what we wanted to do was develop a buffer system for reverse transcription that was incredibly specific for detection of mature microRNAs. So these two systems are now known as HiFlex and HiSpec. They're both in the kit. And you would use the HiSpec system when what you want to do is profile microRNA expression. And this does not work on precursors. It does not work on mRNAs. It does not work on long RNAs. And the advantage here is that it's because it's so incredibly specific when we do profiling, we know that the signals that we get are signals from the mature microRNA, and there's very little downstream characterization we need to do before we move on to focus on the microRNAs that we're interested in. And so this is the way the system works. This is the HiFlex system, and on the left we have the RT kit. We're doing a polyadenylation and a reverse transcription simultaneously in the tube at the same time. We're adding a poly A tail to the targets that don't have poly A tails, like microRNAs and other small non-coding RNAs. And then we're doing a reverse transcription with an anchored oligo-DT primer that has a universal tag on it. So all of our mRNAs, microRNAs, small non-coding RNAs end up as cDNA with a common tag. There's also random primers in the reaction, which lets you use your existing gene-specific primers to look at your mRNA or any of our Quantitech CyberGreen primer assays to look at mRNA. So this is the HiFlex system. So everything gets reverse transcribed into cDNA. And if you look at the bottom here, what you can see, there's a simple setup of a reaction. So 1 hour incubation at 37 degrees. We do a heat kill. And then that cDNA can be used for any mRNA target. So coming back to the question of why you would want to look at precursors, this is a reaction that's been done with the miScript II HiFlex system. And what you see here is a panel of microRNAs and their precursors quantified from the same sample. And you can see that the precursors are generally much rarer. They come up 100-fold to 1,000-fold less abundant than the mature microRNAs, which are much more stable. But there are exceptions, like miR18A on the right side. There's actually quite a bit of precursor that seems to be more stable, and the mature is processed more slowly. The other thing that you can do with this, which is very important, is that because the same microRNA can be encoded by multiple loci in the genome, which are under the control of independent Pol II promoters, you can look at the precursor and figure out which of the loci are actually active. So if you're trying to figure out what's regulating the expression of a microRNA, and there are several of them in the genome, you would use the precursor assay to figure out which one is actually being expressed. The high-spec buffer, as I mentioned, only reverse transcribes very short RNAs, typically under 50 bases or so. This is very, very low background and very high specificity. It doesn't have randomers in there, and it has some other very different characteristics in the RT system. And this is just an example of what happens when you look at HiFlex versus high-spec. On the left, we have 4 microRNAs and 4 housekeeping genes, and you can see the housekeeping genes are very abundant and expressed at high levels. When you use the high-spec buffer, we get no signal at all from these housekeeping genes. So we really suppress the expression of, suppress the reverse transcription of long RNAs, typically by 1,000-fold or higher. When we developed the system, it was important that all of our assays performed equally well with both buffer systems. So this is just the example that everyone shows of LET7, and this is using the high-spec system. And as you can see, when we use different synthetic LET7s and different LET7 assays, we generally see no crosstalk at all. There's some slight crosstalk between extremely highly related sequences here. This is definitely a worst-case scenario. One thing that's important to mention is that we've looked at every single assay that we have for every single micronome that we develop and have done extensive wet lab validation of them. So they've all been looked at for background in total nucleic acid. They've all been looked at for specific products. They've all been looked at for amplification efficiency. So everything that we sell is wet lab, highly validated. Here's an example of our new controls for the miScript II system. Our old controls didn't work because they're designed against targets that worked in the flex buffer, and they wouldn't work in the highly specific buffer. We struggled for a while to find specific assays in these snow RNAs that were close enough to the end that would actually work in high-spec buffer. And what you can see is this panel of snow RNAs with both the high-spec and the high-flex buffer, and they give extremely similar patterns between both systems, and they also are relatively invariantly expressed between these different tissue types. So now that we have a system, let's talk about profiling. And this is the beauty of the merger of SA Biosciences and QIAGEN. SA Biosciences is the premier content development company for qRT-PCR-based profiling. QIAGEN had complete micronomes for screening. SA Biosciences brings both intelligent content, which is on the right side of our page here, the miScript microRNA pathway arrays that are available in human, mouse, and rat for different cancers, general cancer, cell differentiation, immunopathology, inflammation, neurological development and disease, and we'll talk a little bit more about the serum and plasma and MyFinder assays. These are developed by bioinformatics scientists who do a lot of text mining and data mining of the literature to build functional sets of focused content for RT-PCR-based arrays. We also have the most complete human, mouse, rat, and dog micronomes available. We're far ahead of most of our competitors in the assays that we offer in these systems. Again, they're all wet lab validated. And all of these things, since I forgot to mention it, I'll mention it now, are single-use plates. So these are single-use assays that you do an RT reaction, you take a plate, peel foil off of it, and it's ready to use. Every single assay and every single lot of plates are wet lab validated. So we're taking our cDNA, adding it to a master mix, aliquoting it across a PCR array. We're simply peeling the foil off, adding it across the array, running our real-time experiment, and then all of the data analysis, the controls, the normalization, the volcano plots, the delta-delta CT analysis, the publication quality graphs are done with software that's available on the QIAGEN SA Bio website. So you don't have to buy any of this software. It's specific for every array that you buy from us. Even a custom array where you want specific content on the array, you can still do automated data analysis on our website. This is what a 96-well array looks like. There are 84 microRNA assays on this array. Every array has the C. elegans spike-in assay on it for serum, plasma, or any other sample where the SNO RNAs are not expressed. We have a panel of SNO RNAs that we use as endogenous controls. And then we have system controls. We have a reverse transcription control in duplicate and a PCR control in duplicate. So when you run your array, you will know whether your sample prep worked. You will know from sample to sample how much variability you have in your housekeeping genes. You will know that your reverse transcription reactions work properly and there were no inhibitors in your RNA. And you will know that the PCRs work properly. And all of this analysis is done automatically online. We also have the greatest compatibility with instrumentation. So we have specific plastics for the ABI instruments, Eppendorf, Stratagene, Biorad, Roche, Takara, the QIAGEN, Rotor-Gene Q. So if whatever instrument you have, we can produce these arrays to run on your platform, and our software will work with that array as well. And it's also worth mentioning that if you don't want to run these yourself, the QIAGEN SA Biosciences Array Core will take your samples, isolate RNA, do your RT reactions, run your arrays, do the data analysis, and send you back a finished report. So let's talk a little bit about these arrays. The hallmark of these arrays are reproducibility. There's an amazing amount of QC and optimization that goes into producing these things. So what you have here is an experiment that was done by a summer student in our lab who had just graduated college named Krishna, who took 2 HeLa-cell pellets, did 2 RNA preps with miRNeasy Mini, and then did two RT reactions and ran 2 arrays. So there are 2 independent experiments in parallel by a young lady who did her first PCR experiment. And this is what it looks like. She's got 80-some-odd microRNAs on this panel. She's got an R-squared of 0.9862. These 2 samples look absolutely identical. This is the first time that she's ever done PCR, and certainly ever done an array. This is an experiment showing biological reproducibility, again from Krishna, our first-time user. So these are HeLa-cell pellets that were collected from different parental stocks 2 weeks apart, and then they were processed using miRNeasy. An RT was done and the PCR was done. So these are separate cell lines, separate cell cultures, derived from the same HeLa stock in our freezer. And as you can see, perfect alignment, an R-squared of 0.989. This is an example of operator-to-operator variability. So this is an experienced technician in our lab and Krishna, each doing their experiments independently on different days from different RNA preps on the same array. And these 2 different people achieve an R-squared 0.9959. So if you're the same person on different days or you're different people in different labs, these arrays are superbly reproducible. Which brings me to the question about replicates. And we get asked this often of whether people should do technical replicates or not. I can tell you that unless you have a lot of trouble pipetting, the reproducibility of the system is extraordinarily high. And that unless you do something terrible, and you can usually tell that from your controls, technical replicates are really not what you need to be doing. What you need to be doing are biological replicates. So maximize the variability in the experiment and do 3 biological replicates, 3 cell preps, 3 RNA preps, 3 RT-PCR profile experiments, and now you know that the differences you're seeing are true biological differences, and that's what really matters. So instead of doing 3 technical replicates of the same RNA sample, do 3 biological replicates. So let's move now towards doing a little bit bigger of an experiment and doing some profiling. And I'm just going to run through some examples for you in the time remaining. This is a miRNome profile, so it's the whole human miRNome in cultured cells. So these are PC3 cells that are either transduced with an empty control vector or transduced with a vector that expresses p53. And when you compare the delta-delta CT normalized against the internal normalizers, you can see that there's a handful of targets that expression changes when you transduce p53 into these cells. And the ones that are pointed out here, 34A, 940, 203, and 551A, are known targets that are regulated by p53. And here what you see is that they're upregulated with p53 expression, which is what you would expect. And again, this is 2 RNA preps, which is 2 tubes, diluting it, putting it on 2 plates, sticking it in your machine, running it for a few hours, uploading your file to our website, doing data analysis. Here's a whole micronome experiment on a fresh adenocarcinoma versus an adjacent normal sample. And of the whole micronome, what we found was that 451 microRNAs are detectable. And that's about 40% or so. If you set a 4-fold cutoff for strongly regulated microRNAs, there were about 100 that were up or down regulated in the adenocarcinoma. So about 10% of the microRNAs in the micronome were 4-fold or greater differentially regulated in this cancer versus the adjacent normal tissue, which is not that surprising, frankly. Here's an example of treating colorectal cancer cells with 5-azacitidine. And 5-azac inhibits DNA methyltransferase. So what we're doing is we're demethylating promoters and turning genes on, essentially. And what you see in the left panel is untreated on the x-axis, treated on the y-axis, and you can see there's a whole bunch of microRNAs that are differentially regulated when we treat these cells with 5-azacitidine. So there's 104 microRNAs that were strongly upregulated and a handful, 30, that were downregulated. And whether this is a primary effect on the promoter of the microRNA or a secondary effect because some transcription factor was deregulated, it's unknown. But the data that you see here, the delta-delta CT data, the log fold change data, and the volcano plot with p-values were all developed using the online miScript PCR array data analysis tool. So all of these options are available to you with your raw data from your PCR run. This is FFPE samples. The panel on the left are 3 FFPE isolations from lung tumor FFPE that were all run on the same MyFinder array. The MyFinder array is a 96-well array that sort of has the best of the most widely studied and characterized microRNAs. And you can see that these lines completely overlap, that the expression pattern we see from these 3 FFPE samples are the same. When we compare the lung tumor to the normal lung, you can see that there are a couple of outliers that fall off the line that are differentially expressed at a higher level in lung cancer. So I want to spend the last bit talking about serum and the human serum and plasma array. What's on the array are 85 microRNA assays that have been reported to be found at aberrant levels in serum, in cancer, and in other diseases like heart injury, liver injury, atherosclerosis, diabetes. There's a number of housekeeping gene assays, reverse transcription controls, PCR controls, and the C. elegans mimic. The panel on the bottom shows you how we isolate RNA from serum. Our default is to use 200 microliters of serum. We elute from our column with 50 microliters of elution buffer, and we use 5 microliters of that for an array. So 1/10 of our prep is used on either a 96 or a 384-well array, which means that that 200 microliters of serum can be used, if you wanted to, for full micronome in triplicate from 200 microliters of serum. Human, which is 3 384-well arrays. The workflow is to take plasma or serum, extract the RNA, do an RT, do the real time, and then do the data analysis. I think that's clear now. I want to show you a couple of pictures of serum and plasma profiling to give you some idea about what I mean by the stability of these samples. On the top panel here is serum that's either been freshly prepped for RNA or sat at room temperature for 24 hours, and what you see is that it looks essentially the same. On the bottom is plasma that was either prepped immediately or the plasma sat for 24 hours, and it looks essentially the same. So the microRNAs in serum and plasma are stable 24 hours at 25 degrees, as long as you've separated that serum and plasma from the cellular component, because the cellular component would lyse and dump a lot of nucleic acids into the serum and plasma, and you really don't want to do that. Freeze-thaw stability, top serum, bottom plasma. 1 freeze-thaw cycle versus 20 freeze-thaw cycles. The microRNAs in these samples are absolutely stable. You should never freeze-thaw whole blood and expect to get this pattern, because when you freeze blood, all the red cells will lyse and dump a ton of RNA into your sample. So isolate that plasma or serum and then freeze it, and you're fine. If you compare plasma and serum, and this is a question that I've been asked multiple times, this is triplicate isolations. It's 1 person, 3 plasma isolations, 3 serum isolations, all from the same blood draw. You can see that there are a little bit of difference between serum and plasma. The R-squared is 0.92. So the advice that we would give you is pick serum or pick plasma and stick to it, and then you won't have this sort of variation. And the recommendation is to probably use plasma and not let the stuff sit there and clot, so you avoid some cell lysis, you avoid variability in clotting, you collect it in anticoagulant, you spin down the cells, and you take the plasma. If we look at the microRNAs that are expressed in serum and plasma, we find that there are some that are actually very consistent. These possibly could be used as internal controls. I think it depends on your particular experiment, but here's a panel from 9 or 10 normal volunteers where there were less than 2-fold variability between people for these microRNAs. Here's an example of a profile using normal serum samples in panel A. These are 3 different isolations. You can see they all look essentially similar, and this is the serum and plasma panel, so it's 80 some odd microRNAs, and you can see that over 60 of them are detectable in serum, and that they look all the same from the 3 different isolations. When we compare breast cancer serum to normal serum in panel B, what you can see is that most of them line up, and again, they're different people at different ages, but we do see a couple of outliers, and one of those outliers is miR34A, which has been published as a potential serum biomarker for breast cancer. So even in this tiny experiment, we can actually see the same data that was seen in this publication. So I'd like to spend the last minute or 2 talking about data normalization. So we're all familiar with using GAPDH and cyclophilin and actin and SNO-RNAs for data normalization. That does not work in serum and plasma. So the first thing we do when we do a profile on one of our arrays for serum or plasma or other liquid samples like urine is we look at our controls. And here you see from 2 samples the RT, the reverse transcription control, and the PCR control, and you see a raw CT value for sample A and sample B, and you can see that these values are extremely close to each other. These are run on different plates. Right, so sample A and sample B are different runs. And there's smarts in the software that tells you whether these numbers are good or not and that your array worked. So now we know our arrays worked. What do we do now? Well, these are the housekeeping lows that are on our array, and what you see are CTs of 35, and that essentially is non-detectable. So they're variable, they're very expressed at very low levels, they're not dependable as normalizers, so what do? Well, so the suggestions are, one thing that you should do is spike in the C. elegans miR39, and that will at least tell you whether you've got gross differences in the recovery, the raw recovery of RNA from your samples. Did your technician lose half of one sample and not half of the other sample? And that's good for a first step in normalization. The other things that you can do is you can normalize the data to the geometric mean of the expressed target. So if you're screening a plate or a miRNome, you can normalize to everything. essentially, for targets that are expressed, targets that have CTs less than 35. Now, if your samples are very different from each other, you may have different numbers of expressed targets on the different arrays. And so a better way to do it would be to take the targets that are expressed, commonly expressed in all of your samples, and get the geometric mean CT of those and normalize against that. And that works pretty well. Now, and it works pretty well as long as you have enough detectable targets, as long as the samples aren't really different from each other, and that there's only a few microRNAs that might be differentially expressed. The final way that you can do this is to use a program such as Norm Finder, which is a publicly available tool. And what Norm Finder can do is it can suggest which microRNAs in these panels are the least variant in your experiment and which ones you should try to use as normalizers. And what we typically do is we try that, and we try the normalization to the CT, the mean of targets that are commonly expressed. And generally, the data comes out the same. Occasionally, microRNAs that are slightly upregulated come out slightly downregulated, but the larger shifts that we see are robust and survive these different methods of normalization. So finally, I want to wrap up with one slide and mention the GeneGlobe website. This is at QIAGEN.com/GeneGlobe, and all of our gene-specific content lives on this site, and you can find all the microRNA products here with the exception of the arrays. So at the moment, if you're looking for the miRNA arrays, that would be found on the Assay Biosciences section of the QIAGEN website, and that's also not too difficult to find. And if you're really lost, give us a call and we'll tell you where everything is. And with that, I want to thank you, and I want to remind you that this was the third of a set of 3 webinars. A copy of the slides from each of these webinars is available to you, and the webinar recordings will also be available on the website. So if you've missed any of these, you can go back and listen to them. And I also, one last thing before I thank you is to remind you of my email address if you have any questions that are not addressed in the remaining few minutes. And with